this video we will discuss about the role of this COMMD3 in the pigmentation, or in other way in the, we can say in the melanosome. So what they have studied is that COMMD3 helps in maintaining the neutral pH of melanosome that results in tyrosine functioning, and that finally helps in that finally provides the pigmentation of the cells. So how they have concluded come to that conclusion, we will discuss this through this video. So uh, in the first in the first panel, or we can say the panel A, represents the immunoblot experiments. So first one is the input part is the whole cell lysate. In this they have identified whether it has these melanosome markers, and it has they have also identified that it has this COMMD3 and this mitochondrial marker. After that they purified these melanosome proteins and do a co-immunoprecipitation assay against these two, melanotag MC and melanotag HA, and they have expressed this melanotag HA in those cells, and in this they have find out that this melanotag HA is present, and its result is acceptable as it does not have any mitochondrial marker, and it is purified as and it is also purified as it has all the it has these two melanosome markers, whereas when this melanotag MYC co-immunoprecipitation experiment was done, we do not see any signals neither for mitochondrial marker nor COMMD3 and nor for any melanosome markers, because cells are not expressing this melanotag MYC. Cells are expressing this melanotag HA in this experiment. Next, they check the pH through this LysoTracker red, and the more red the color is, the more acidic the pH of the sample. So when they check it and compare it with the wild type, they can see that in the case of this COMMD3 knockout, the cells are more red in color. That indicates that it is more acidic, and they verified this result using the flow cytometer also. All the COMMD3 knockout has high uh, intensity of the red signal, indicates that they have high acidity. And when they did uh, when they uh, regrow the COMMD3 in those cells, they can see that decrease in the pH, as we can see in the images. However, instead of COMMD3, if they supply another proteins that is not related to the that is unrelated or not related to this melanosomes or pigmentation process, but are dysregulated when this COMMD3 is absent, COMMD3 is knockout or absent. So when they supply this RAP2A, they can see that there is no effect in the acidity of the pH. So this further made their point that COMMD3 helps in maintaining the pH. And later on they try to establish the pH through different ways, that is using this BAF-A1 and CON-A, and when they add these two, they are able to see that the signals are restored and almost to the control level. So from this they made a point that COMMD3 will not affect COMMD3 will play a role in the maintaining the pH of this melanosomes. Or then thereafter they also check the melanin content in these different knockouts, and they can see that in whenever they introduce this BAF A1 con A1, they can see the increase in the melanin content, or the pH is restored. And they also used one more one more case in which they introduced chloroquine. So chloroquine also helps in restoring the pH, the endosomal organelles such as lysosome and melanosomes, and they can see that the pH has also been increased. And in the wild, and they are also comparable to the wild type. A vehicle is the case in which it is the COMMD3 is knockout, and in this case the wild type vehicle is used, in which no injection of this, no supplement, no supply, no supply of this either BAF1, CON-A1 or chloroquine is provided. So we can see that the chloroquine levels are restored whenever the pH is restored. And so what actually has happened whenever this COMMD3 is absent, is shown in this panel G. So in the normal case, the tyrosine is active. So tyrosine is active as this ATPase helps in controlling the pH of these melanosomes. So the pH remains neutral. So this tyrosine is active, and it can helps in the pigmentation, or it can provide the tyrosine can do the normal function of the pigmentation. However, when this COMMD3 is knockout, what happens in this case is that the ATPase ATPase is not able to perform its function, as COMMD3 helps in regulating the function of ATPase. So because of that, what happens is that the pH of this tyrosine gets reduced, and that makes pH of these melanosomes get reduced. That makes the tyrosine inactive, and thus no pigmentation effects is shown. No pigmentation effects is seen in this COMMD3 knockout. And thereafter, what they did is that if we somehow we can regulate or control this ATPase regulation using some extra some exterior effects apart from this common D3 knockout, such as using this CON-A or conmycin A or BAF1, then we can restore the pH within the melanosomes, and the tyrosine again gets active, and it will further starts to show pigmentation, or it will further start to show pigmentation or behave like a wild type melanosomes tyrosines. So I hope you have understood the relationship between how this COMMD3 regulates pH of melanosome, and that in turn makes the tyrosines functions, and that in turn makes the conditions in order to uh, in order to uh, in order to make tyrosines uh, behave in a normal manner or behave efficiently. If this COMMD3 somehow gets knockout or non-functional, then the pH of these melanosomes get disrupted. That results in tyrosine inactivation, or it will not behave normally, and that is reflected in their final outcome, that is pigmentation of the cells, pigmentation of the samples. So if you need to understand this COMMD3 and how this COMMD3 regulate pH of the melanosomes in detail, or if you want to understand this study in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.